All right, everyone. Thank you so much for coming. It's great that you're here. You're in for a treat. Um, my good friend Doug LaRowe is here. I've known him for at least 23 years, maybe longer. We can't really remember. Um, so it's really my pleasure to be able to host him here and to have experienced, I think, his, I would almost say, rise to stardom. Like he has done a tremendous job over the past couple decade or so in really being a prolific writer and having just great high impact publications that are really, I would say, somewhat renowned in people who are interested in carbon cycling. So it has really been my pleasure. I'll just read you some of his CV here because that's what I'm supposed to do. He is currently an associate professor and Claude C. Albritton Junior Chair in Earth Sciences at Southern Methodist University. This is an endowed position. He has a bachelor's of arts in Earth and Planetary Sciences from Washington University in St. Louis, a very famous place in terms of space science. He has a PhD with Hal Algeson. If you don't know that name, you should ask Doug about it and all the stories involved in this person at the Earth and Planetary Sciences at UC Berkeley. He then did a postdoc at Utrecht, was a research scientist at Georgia Tech, and finally had several years at University of Southern California, where he had various titles, ultimately leading to a research professor position. If you didn't read the advertisement that I sent around, his research is focused on quantifying how life is bounded by energy and time on Earth and beyond. A central theme is quantifying the thermodynamic potential and rates of reactions that microorganisms catalyze in natural settings and in the lab. He combines computational modeling, theory development, fieldwork, lab analyses, microbial work to better understand the limits of life and how microorganisms impact their surroundings. From his website, his research areas include geochemistry, astrobiology, environmental microbiology, organic matter, bioenergetics, thermodynamics, carbon cycling, and hydrothermal systems. So the work that he does with the bioenergetics and modeling is really centered around all of these disciplines. And so I think that is a great advantage and a great opportunity for Doug over the years to have been really at the heart of a lot of these great research. So with that, I'll turn it over to Doug. He's going to talk about a very easy topic, carbon cycling and natural organic carbon. So thank you, Doug, for coming. Welcome.
Thank you for that introduction, Roy. Thank you for donating an hour of your Friday to me. I'll try not to waste it. Before I came here, a few days beforehand, I had to email Roy and ask him what title I had given him, because I had no idea. And this is it, Unraveling the Complexity of Natural Organic Carbon. And I figured I'd start the first slide, right, and show you about the complexity of natural organic carbon. So this is a mass spectra. This is mass, basically. This is intensity. And this is showing a way of analyzing the organics in natural waters. This is from the Stony River. And you can't see this from the plot, of course, but the authors claim that they identified 36,000 organic compounds in the sample. All right, this is complicated for sure. This is the complexity part. But what I normally like to talk about and what I normally use as my title for presentations is this, life's limits, because I can say anything I want then. And I don't have to remember my title, like I had to email Roy for this one. So I also like to talk about life's limits and how organic carbon is involved in setting that. So mostly what I like to think about is that microbes like to eat organics. So that's kind of the theme today. All right. So the first part of the talk, there's going to be two very distinct parts of this talk. One has dark background for the slides, and the other one has a white background. So if you take a nap and you wake up and the slides are white, you are in the second half of the talk. All right, so here's the first half, basically. Imagine we have seawater. We've got a boat up here for scale. We've got seawater, and we've got sediment below. There's a flux of organic carbon that gets deposited into the sediments, and then the microbes are very active down there eating these organics at different depths and over different timescales. All right. So the questions that I think about related to this is, at what rates are these microbes active? How deep are they consuming this organic carbon? And finally, the really important thing, does this mean anything? Should you care? I guess it depends on what you're interested in, but. All right, so to get you all sort of on the same page, many people here know about marine sediments, but I just wanted to remind you the colors are coming out very strangely.
Welcome, so.
All right, I can't see colors very well, so now I don't know what my.
It's shallower near the land.
Thank you. So this is thickness of sediment, as it says down here. Okay, so most of the planet's covered in sediments. The thickness varies substantially. We're looking at orders of magnitude, up to many kilometers thick near the coastal areas, especially if you look up in the Arctic. So there's lots and lots of sediment on the planet. We all kind of know this, but I just wanted to refocus your mind on this topic you may have encountered years ago. Okay, so a bunch of people have gathered over various years cell concentrations as a function of depth in these sediments, and they've determined that there's about 10 to the 30 microbes in marine sediments, right? So this is a very, very large number. I'll be covering about 50 orders of magnitude today, and this is on the high end. I'll be going very, very small after this. So the idea is that there's a tremendous amount of life in marine sediments, right? Okay, and just to give you a sense of scale for the number, if there's 10 to the 30 microbes, there's about 10 to the 24 stars in the universe. So there's 1 million times more microbes in marine sediments than there are stars in the universe. So big numbers. All right. Okay, and depth-wise, we know that these microbes can be found at least 2.5 kilometers beneath the seafloor. So this is looking at cell concentration. This is in cells per cubic centimeter in a logarithmic scale as a function of sediment depth. And these dots are just cell counts people have done. And you can see there's some jumps in the concentrations in these organic rich areas, these coal beds. And they found microbes down to 2,500 meters below the seafloor. Okay, people have been able to take microbes from very old sediments, bring them back to the lab, and grow them. And these are just showing a couple of slides. People from this is 101.5 billion-year-old sediments. They were given inorganic carbon and nitrogen labeled, and organic carbon and nitrogen that was labeled, and they incorporated these nutrients and grew. So we have evidence that even microbes that have been sitting around for 30 million years before the dinosaurs went extinct can be resuscitated in the lab. All right, so that deep life, that old life is capable of doing something. And another thing I wanted to make a point about is that these microbes, these single cells in the marine sediments and the subsurface in general make up about 85% or more of all the single-celled organisms on the planet. I should specify bacteria and archaea, not just single-celled. So we have a lot of our single-celled prokaryotic life buried in the subsurface. So this is why some people might want to care about life in the subsurface. And most of it is down there, at least most of the single-celled life is down there. Maybe this is the normal way of living for these organisms. And we think of the surface world as normal because we're familiar with it, but it turns out the subsurface has a lot of life in it, and maybe we should study what they're doing to get a sense of what's possible for life. Okay, so again, my questions are, at what rates are they active? How deep are they? And does this mean anything? Okay, so a nice thing that people have done over the years is they've gone out to sea on a boat, big boat, and they've drilled through the water, go through the water column and go into the seafloor, and they bring back these wonderful sediment cores. And you can, of course, think of this as a way of, you can measure this as a function of depth, but I like to think of them in terms of time, right? These are gradually being accumulated year after year, and they're showing us chronology of an environment. And since we know time, then we can say something about what the rates of the activities are as a function of this depth into the seafloor. All right. And of course, when they take these core samples, they're not just looking at how old are they, but they take a lot of different subsamples throughout as a function of depth. And people have measured all kinds of things, like they've measured the cells, they've looked at the chemistry of the sediments, they've looked at the mineralogy, pore fluids, lots of different things have been measured, which is wonderful because I like to use other people's data. I'll be generating my own very soon, and that'll be a conversation for maybe lunchtime. Okay, so what I do with all these measurements, and this is a global set of measurements that people have been doing for many decades, is to basically take all these measurements and stuff them into a black box called a reaction transport model. And so what this model can do is it takes into account this accumulation of sediments over time, and we can look at rates of reactions as a function of depth in these sediments, right? And then we can say, basically, because we know that it's microbes that are catalyzing these reactions, that's the level of microbial activity in these sediments. Okay, and so one of the key things we can get out of this information is if the rates of organic carbon degradation and microbial power use. So this is sort of getting at this unraveling the complexity of organic carbon, and this is getting at sort of limits of life. You may have thought about or maybe heard someone talk about thermophiles or mesophiles. There's all these different philes out there, and these are groups of microbes that can thrive under those conditions. I like to think about microbes who can thrive under very, very low energy and low energy flux conditions, right? So this is not typically something we think about with microbes as a particular survival strategy, but I think it's crucial to understanding the subsurface is you have organisms that are through evolution designed to withstand very, very, very long periods of time where almost nothing is happening, but yet they're staying alive. In fact, my colleagues Drew Steen and Karen Lloyd have tried to coin the term aeonophile as an eon, as lovers of time for this type of microbe. We got in trouble actually because we were having a conversation about what to call them, and we thought we should call them chronophiles, like chronos for time. It turns out that word is already in use in some fields of psychiatry to describe people who have an abnormal obsession with young people. And we got caught just in time before we published that word, and that would have been really terrible. So, what are these microbes doing in the sediment, right? So they're in this sediment column, and I'm going to a more of a schematic version of the sediment column now. We have water on the top, basement at the bottom. So these microbes are eating organics, right? And that's a pretty complex organic right there. But as we know, there's lots of different types of organic compounds. On the very first slide I showed you is from just one river sample. They identified over 36,000 of these compounds. You go into those sediments and you've got probably a lot more than that, right? And there's a wide variety of organic properties. And the way I like to think about this is here's two types of organic carbon. So here's ethanol in vodka, and this is Kevlar, the thing that can stop a bullet in a police vest, right? Organic compounds. These have very different properties. Sometimes they're related, depending on what happens, but organics nonetheless, right? So do we treat them all the same? Probably not a good idea. You wouldn't want to have a bullet flying at you and you hold up your bottle of vodka to stop it. Okay, so what's happening? So there's organic material coming through the seawater, it gets deposited on sediments, and then as you get deeper into the sediment column, the concentration of organics typically goes down, especially at the very top, right? So we want to be able to distinguish between ethanol and Kevlar in the sediment. Very different properties. Okay, so traditionally what people have done when they do this reaction transport modeling in sediments is they consider that there's a pool of organic carbons, all the organic carbon that's there, and it has a characteristic rate constant. Let's call that K1. And you can easily calculate how the organic carbon concentration changes with depth just on a very basic little differential equation like this. And then people realize, like, well, this doesn't work all the time. We need to have a more sophisticated approach to quantifying organic reactivity. So they said, there's two pools of organic carbon. And one pool has the K1 rate constant, like above, and the other part of the organic carbon has a different rate constant. And it can be 50-50, or usually it's not. And then you add the second term to your little equation here that characterizes how the organic carbon is changing over time. Well, that, two pools of organic carbon to describe 100,000 compounds globally is not sufficient. So the approach that I've used with colleagues is something called a reactive continuum model. And what it simply says is there's an infinite number of pools of organic carbon. Just saying that there's a lot of different kinds of organics, and the distribution of those changes with depth and time because the stuff that's very reactive goes away quickly, and the stuff that's not very reactive goes away over longer time scales. So you can assign either an infinite number of reactivities, or like we did, we chose 500, which is close enough to infinite for organic carbon to capture the complexity of organic carbon in marine sediments on a global scale. Mathematically, it looks more complicated, but it actually ends up being simple to put into an analytical solution, so we don't have to do a numerical scheme. It makes computing time much, much shorter. Okay, so that's how we characterize organics. again, coming back to the questions, I know people check phones and think about their mortgage and they're doing other things during a presentation. So I like to come back with questions over and over again. At what rates are microbes active in these sediments? How deep are they? And again, does this mean anything? All right, so let's look at some results. This is like 3 papers condensed into one slide. So what we effectively did was we said, okay, we divided the sediment columns globally. This is in a 1/4 degree by 1/4 degree scale. And we said, okay, there's a bioturbated zone at the top. That's where you've got snails and things that churn up sediments. And the rest of it below there, we call this the rest of the Holocene, basically. So the non-bioturbated part of the Holocene sediments. And then beneath that, we have Pleistocene. And of course, the basement's drawn here, but that's not necessarily the case. The Pleistocene only goes back 2.6 million years. So I'm not saying that we're only considering sediments that go to the basement of the Pleistocene. This is just an illustration. So what I'm going to show you is the rate of organic carbon degradation in this zone right here, sort of this non-bioturbated Holocene section, just as an example. There's a whole lot of plots like this in this paper. And so what you're seeing here are grams of carbon per cubic centimeter per year globally in this depth integrated organic degradation rates. Again, this is just the bulk of the Holocene sediments. And the first thing to note is there's about 5 orders of magnitude difference here, right? The globe's a big place. In some places, the organic carbon is being degraded very rapidly, as in these Arctic areas again, in the coastal shelves. And of course, in the open ocean, where you have a lot less organic carbon, the rates of degradation are much, much lower. All right, so but we have this sort of applied in 3D to the whole globe, but I'm just going to show you this one example of these rates of organic carbon degradation. All right, so that previous slide took into account bathymetry, porosity, and sedimentation rates and organic carbon concentrations at the sediment water interface, and on and on and on. All these global data sets that lots of other people have gathered over many, many years, all packed it into one MATLAB code. So now I want to add to that distribution of cells, which again, other people have gathered and made global sort of projections of where they haven't measured them. And also my own sort of component, what I always like to do is calculate the energetics of stuff, right? So we want to look at, okay, we know the rates of organic carbon degradation. So what about how much energy are these bugs getting from this process? All right. So what we did was to know how much energy they're getting, we have to know what reactions they're catalyzing. And the bulk of the organic carbon in marine sediments basically gets degraded through 3 different pathways. And one is through oxygen plus organics. The other is sulfate reduction. And then there's a bunch of organic stuff that gets turned into methane. The methanogens are happening down there. And it usually comes in this sequence with the oxygen's at the top, the sulfate's in the middle, and the methane's at the bottom. Now, we don't a priori know that this is the case everywhere all the time, but luckily some other colleagues have determined the depths to which oxygen penetrates the sediments and the location of this interface right here. And so these are some global data sets that we re-massaged to put into our model. And this is showing global oxygen penetration depth. So you can see in some sediments, like in this red color here, the gyres, the ocean oxygen penetrates all the way down to the basement, more or less. And in lots of other places, oxygen only penetrates a millimeter or so into sediments, right? So again, you have to think of the sediments as being very different in different locations. So we're taking this into account. And so another set of colleagues measured and determined the sulfate methane transition boundary globally, and we stuffed that into our model. So now we have the depth at which the sulfate methane transition takes place. Where it's white, there is not a sulfate methane transition, either because there's no methanogenesis or no sulfate reduction. So you can see here, this depth of this goes up to 1,000 meters on this slide. So we have this spatial distribution of which of the reactions are taking place where. So this is, again, where you're going to have the organics being processed in different ways. And so what we can do is say, okay, in this zone here, we have oxygen plus organics. And in this zone, we have sulfate plus organics. And down here, we've got organics becoming methane. Those are 3 major sort of catabolic zones. Okay, so we can calculate the Gibbs energy of this reaction and of that one and of that one. Three different energies. And we know now from that slide I showed you that has 3 years or 3 different papers collapsed into one. We know the rates of organic carbon degradation as a function of depth in 3D globally through the Pleistocene. So we have that. And what we can do with that information is put this into units that sort of tries to understand what is the least amount of energy a microbial cell can get by it. What's its like lowest, right? So that's a limit to life. And I put this in units of power. So if you're not used to thinking about power as energy per time, a watt is a joule per second, right? So this is joules per second per cell. All right. So here is a result slide. I'm going to show a few of these real quick. And again, this is a heat map. So this is looking at the integrated power usage per bean cell in the oxygenated layer of sediments globally. All right. And so the scale, these are really small numbers, but I'll do some comparisons later to help fill in the gaps in your thinking here. So you can see some of these red numbers. Most of it's kind of red, yellow, and orange. So 10 to the minus 16 joules per second per cell, 10 to the minus 18. You don't see much of these blue colors here. So a bit higher power utilization in this oxygen zone because you get a lot of energy from using oxygen, right? Okay, so let's look now at the sulfate producing zone globally. And so this again is the integrated power usage per bean cell. And you can see a lot more colors on this map. So there's a broader distribution of powers being utilized by the microbes from 10 to the minus 16 in some areas, and 10 to the minus 21 or so in other areas, right? So we have a different amount of energy per reaction and a different rate at which it's being used, both. And finally, we get down to the methanogenic zone. And you can see, first of all, a lot of the sediments are white. And that's because there's no methanogenesis in these locations. But where you do have it, it tends to be in these green and blue colors. So the power output is 10 to the minus 19, 10 to the minus 21 or so. All right, so now I'm going to take all these calculations and I'm going to add up all the cells and show you how they fit in a distribution of their energetics. All right, so this is all these microbes in the Pleistocene and Pleistocene age sediments. So this is cell abundance here. This is times 10 to the 26, another big number. And this is their power per cell of all these. And so the methanogens are sort of this peak here. The colors really came out funny. The sulfate reducers are sort of in this distribution, with this little bump over there. And the oxygen utilizers are definitely in the higher end, but again, they all overlap with each other. So knowing what they're doing to get energy doesn't tell you a priori what kind of power utilizing they're doing. So a lot of the oxygen utilizing microbes are out in the abyss, and they're actually very, very slow. But they're still breathing oxygen. And some of the sulfate reducers are very, very fast because there's a lot of organic matter present. They can generate a fair amount of power, more than oxygen in some cases. So this is just sort of some really small numbers, and I want to run through a comparison real quick of how this stacks up to things you might be more familiar with. So I extended the scale all the way down to 10 to the minus 10, up to 10 to the minus 10. So the lowest known power utilization rate in the laboratory is 10 to the minus 15 watts, right? So basically, all the cells in marine sediments are operating at orders of magnitude less power than the lowest known one in the lab. Okay, so we all knew the subsurface biosphere was probably going to be slow, but now we've quantified in a way how slow are they? What rate are they getting energy? Over here, I've got 10 to the minus 10 are the neurons in your brain are operating at this level. Okay, even if you're asleep right now during my presentation, your neurons are still combining glucose with oxygen and generating about 10 to the minus 10 watts, right? And overall, your body's about 90 watts. So you're all operating about 90 watts, just to give you a sense of scale. So these bugs are living at 10 to the minus 20, minus 19 watts. Of course, they're single cell, right? And one thing I really want to illustrate here is that it seems that all these numbers trail off and disappear at about 10 to the minus 21. And I gave this a special term, a zeptowatt. So if you go look up your SI prefixes, zepto is 10 to the minus 21. And I was overjoyed when I saw this. I got to use zepto in scientific publication, and I'm still hammering at it. I still say zepto all the time, just like I am right now. So this seems to be a power limit for life. We know a temperature limit's 122, minus 15. We can say pressure limits. We can talk about all these different limits of life, pH, radiation dosage. here's a power limit for life. How little energy can they get by? Okay. So the whole idea here is to take the complexity of organic carbon into account and say something about life's limits. So this is kind of what I've focused on in the first half of this talk. And you could possibly think through, like, what is a ramification of this? Well, we think about if we're going to find life somewhere else in the solar system or beyond, it's going to be in the subsurface, because in our solar system especially, the surfaces of planets are terrible places to live. They're irradiated, they're cold, they're hot if you're on Io. You're going to be in the subsurface if you're going to find life. We're going to find life in these places. And now we know that organisms can be sustained over tens or 100 million years on ultra low fluxes of power. So they don't need a lot to stay alive. Okay, so now this slide is here to tell you that I'm jumping into the second half of the talk that is going to focus more on this unraveling the complexity of organics. And like Roy said earlier, I do calculations for a living, but I'm going to show you the results of experiments. I didn't actually do them, but I helped with the design and the ideas behind it. So if you get into nitty-gritty detailed questions, I'm probably not going to be able to answer them, but I'll do as best as I can. Okay. And to frame the second half of the talk, again, we have the white background now, new part two. One way to take all the complexity of organic carbon and think about it with one number is to consider its oxidation state. All right. And you're familiar that carbon dioxide has an oxidation state of +4, methane's down here at -4, and organic compounds fall all the way in between, all over the place, right? And I just have a few examples up here. Here's formate at +2, glucose at 0 or neutral. We have an alkane, a C16 alkane down here, a little below -2. And they fill in, you can put in all the different thousands and thousands of organic compounds, and they'll all fit on this scale. And so you can then refer to this massive complexity of organic carbon with a single number. Very very simplistic of it, I know. And why would you do that? All right. So why do we care about having a single number? Well, again, we can taking a step back. You can, if you have the elemental ratio of carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur in the charge, then I can calculate this oxidation state of organic carbon, right? So I won't go through the formulas for that. You can get one of my old papers and look it up. And the nice thing about that oxidation state is it correlates with the amount of energy you can get from oxidizing that organic, right? So you might have 36,000 compounds in your sample, but in that FPICR analysis, you don't know the form, you don't know the structures of them or anything else, but we can correlate their formula to the oxidation state, calculate it, and then use that to get an assessment of how much energy is available from eating that organic, actually from oxidizing it. Okay, so to sort of show how this works, this is an updated version of a plot I made in 2011. So this is a paper that was accepted days ago. So this is showing, looking at this reaction here, we have organic compounds being oxidized to CO2, some electrons. So it's just a half reaction. There's electrons there. And this is a wide variety of types of organics: alcohols, amides, amino acids, aromatics, ketones, nucleosides, organic acids, organic sulfur compounds, oxocarbons, huge number of different carbon sources. And if you write this reaction right here and calculate the Gibbs energy of that half reaction, this is just a schematic, then you get a trend with the oxidation state, which is down here. These are reducing compounds, oxidizing compounds. You see this trend that the more oxidized organics, the Gibbs energy is going more negative as you go up, so they yield more energy than the things down here. So there's a pretty good correlation here. This is probably 150 or so compounds. And so this is what we're going to be, this is sort of the idea behind why all the experiments I'm going to show you were done. The idea that the energy content of the organic carbon is going to be deterministic about what happens to an experiment. All right. Okay, so here's what we did. So this paper just came out earlier this week. Two colleagues at Lawrence Livermore Lab in Nature Communication. Maybe it's not out yet, but it's accepted. And so here's what we did. Who did? He took rice paddy soils as the inoculum and took a bunch of different individual organic compounds of different oxidation states. Now, we need a lot of different ones, and I'll show you what they are. But the idea was to get a broad spectrum of oxidation state, a broad range of structural classes, and the kinds of compounds that are actually found in this kind of environment. right? So we wanted to cover what's naturally there, a broad variety of not just oxidation state, but structure as well. Okay, so we combine those and put those in a bunch of test tubes, all right, and 96 well plates to do some microbiology stuff, and incubated them for 190 days. All right, so rice paddy soil, organics, 190 days. And then measured a whole bunch of stuff, including CO2 and methane that came out of the incubation experiments, the microbial biomass, the 16S for phylogeny, metagenomics work, and energetic stuff as well. And I'm not going to show you all the results. There's tons of stuff, but I'm just going to hit some of the highlights here. Okay, so this one is, again, the red colors are going to correspond to the more oxidized organic carbon. The white is kind of the neutral, and the blue is the more reduced. So lots of these different oxidation states. And one of the first things to look at is, okay, does the oxidation state of the organic carbon you give this experiment determine the ratio of CO2 to methane produced? And the answer is yes. So this is the amount of methane produced of the total carbon. So you can think of this as a lot of methane being produced and very little. And this is the oxidation state of the organics that were given to the system. So what you can see is this is organic acids, aminos, sugars, methylated compounds, amino acid mixture. Direct substrate refers to compounds that methanogens can directly use. They don't need to use, they don't, like some of these things like sugars have to be fermented before the methanogens can use them. So this is a substrate like acetate, methylamines, etc. And so what you see is, if you look at just this color dot and this line right here, those are just the amino acids. So they just span +1 to -1, but they have a very strong relationship between how much methane is produced from those compounds, depending on the oxidation state of the organic carbon. And all the other ones also have a trend where, again, the lower the oxidation state of the organic carbon, the more methane it's produced from that inoculant. All right. So if you're fertilizing your fields with reduced organics, you're going to generate more methane. Okay, so now a quick look at how this changes the microbial ecology. And I have a whole lot of slides on this, but I'm just going to show one. And that is to look at what's called the Shannon index as a function of the oxidation state of these organics. So if you don't know what the Shannon index is, it's a measure of microbial diversity. So again, we looked at the microbial diversity before and after the experiments. And when you give them more reduced organic compounds, you have a more diverse community. Now, we also did metagenomic work, and there's a whole other story there I don't have time for. But you see the trend again, the amino acids alone has a very steep function of this oxidation state of the system. And all the compounds taken together, it's not quite as steep, but still there is good correlation between oxidation state and diversity of the community. Okay, now, so yeah, lower oxidation state, more diverse. Okay, so now I want to look at how that oxidation state influences the reaction rates, right? So this is going to be measured by the half-life of the carbon left over. So again, this is a half-life, the number of days the organic carbon remained in the system. Again, this is the oxidation state of the organics that were given it. And as you can see, that the more reduced carbon stays in the system longer. The more oxidized stuff, which is more energetic, is eaten faster. And this is what we'd expect. The microbes are going to eat the stuff that gives them more energy first. All right, so higher oxidation state, more reactive. All right, so now we're going to combine this sort of looking at these different oxidation states with reaction rates and the reaction products. This is a bit more complicated, but I try to white out as much of this as possible to show it. So this is measured carbon dioxide versus methane in these experiments, and this is time, again, it's 190 days of the experiment. So this is at first, I'm just going to show you giving it two different amino acids that have different oxidation states. So serine here is more oxidized, the oxidized amino acid, and this is how much CO2 was produced throughout the course of the experiment. All right, and now I'm going to show you how much methane it produced. It produced less methane throughout the experiment. All right, and it was slower. This took off a bit slower. So again, so the oxidized organic, more CO2 versus methane. And if you give it leucine, which is a more reduced amino acid, it's inverted. More methane is produced than CO2, but the CO2 is produced faster. These are the CO2 samples. So it starts to produce CO2 quickly, but then in the long run, more methane is produced. So that's a bit more complicated to think about, but again, you have basically high oxidation state of the organics, more carbon dioxide is produced, but it's faster CO2 than, well, that should be And the methane is made faster as well from a high oxidation state. So methane production is fast relative to the low oxidation state organic. And the low oxidation state organics produce more methane in general. Now, I'm going to lump together a whole bunch of these compounds, similar experiments, but instead of putting it in methane versus CO2, I'm going to put it in greenhouse potential, right? You can convert methane into a CO2 by how much more heat trapping it is in carbon dioxide. So here is looking at CO2 equivalent. So think about this as greenhouse forcing right here. And again, this is the time. And so this is the oxidation states of a bunch of different organics that were given. This is, again, a range of amino acids, from these reduced blue ones to the oxidized red ones. And what you see is that even though the ones that were given these reduced compounds, they're slower. You can see that they're slower to start producing these CO2 equivalents. But in the long run, because they produce more methane, they produce more greenhouse forcing. So they're actually worse for the environment in that sense. And the ones if you give them oxidized compounds, like these red ones, they start metabolizing faster, but they convert more of it to CO2 than methane, so it has less of a greenhouse forcing implication. All right. So that's kind of like we're trying to make sense of like there's this under theoretical prediction about how microbes are going to respond to different oxidation states of organic carbon. And here we have evidence that it is determining the composition of the community, the rates of CO2 and methane production, and the total amount of methane produced. I said that twice. Okay, so that's just sort of the higher oxidation state, faster CO2 and methane, lower oxidation state of organics, more greenhouse forcing in the long run. Okay, so the basic gist of these set of experiments are, let's take a bunch of fermentative methanogenic communities, give it different oxidation states of organic carbon. So you give it these positive things, these oxidized organics. You have fast responders, and there are specialists that pop up. The metagenomics showed this, but I didn't get into that. But you have these specialists that are the fast responders, and they generate more CO2 than methane. You give them reduced organic carbon, they're slower, but they make more methane in the long run. Okay, so if we had done this experiment for a shorter time span, we would have gotten different results. We had to wait for them all to plateau out, tell us how much CO2 and methane they were going to generate over time. Okay, so I would like to acknowledge collaborators. This is the first half of the talk, people. Got that in the dark colors and the white colors. This is the collaborators from the second half of the talk. And there's some funding agencies at the bottom. And so when you talk to your grandmother tonight and you have your conversation about what did you see today at the time of this presentation, you learn about what a zeptowatt is, and that's a power limit for life. And yet the oxidation state of organic carbon can determine how a microbial community is going to behave. Thank you for your time.
One question from that.
Yeah.
So much. I was wondering, like, if you compared the like microbial communities and the chemicals, the organic compounds available like in marine sediments versus terrestrial sediments, and if that varies much, and also how that might like look in sediments that like in terrestrial sediments that add like marine sediments layered in there in an area that used to be marine, or an area that's now marine that used to be terrestrial.
So that slide I showed was demonstrating the depth to which microbes have been found in marine sediments. There's 2 coal bed layers in there.
Okay.
And that used to be terrestrial. So in those areas, you'd have the complex sort of polyaromatic organics you associate with coal or in those layers, and the cell numbers were higher. And in fact, they looked at microbes that were in those coal seams versus the marine sediment sequences. They're very different communities. There's huge differences between basically freshwater and saltwater bugs. And that's very discernible. Even after all those years of sedimentation, there still is a distinct terrestrial community of microbes in the terrestrially deposited material. In terms of the kinds of organics that you find in marine versus terrestrial, that's what I'm building my lab to do. I want to measure the oxidation states of organic carbon and take into account all the things that are going to make my samples hard to analyze. Because when you look at the CHNOS of a sample, you're going to get stuff from minerals, and I want to exclude that. And from a conversation I had earlier today, you could get microplastics that'll screw it up. So there's all these different things that are going to mess up my analysis, but that's what we're going to try to figure out. And also the FTIACR stuff, where we showed the 36 different thousand compounds from fluids. There's a lot of analysis like that that's done with different sample types. It's a little hard to interpret what it all means because you get thousands and thousands of compounds, and so they plot in different spaces, CHO space. So they're different, but yeah, I'll stop that. Yeah.
So I had a question about the sort of redox sequence that you showed in the beginning.
Yeah.
So I was taught like that it's like oxygen first, then nitrate, and then manganese and iron,
Yep.
and then sulfate, and then methane. So I was wondering how come you didn't include like nitrate and the metals?
Two reasons. One is they are relatively trivial global significance when it comes to degrading organic carbon sediments. Lots of other people have shown that the three pathways I showed account for the vast bulk of organic carbon degradation. We also don't have a great way to say like where is the nitrate zone. The other ones we have oxygen penetration depth, we have the sulfate methane transition zone, globally speaking. But for those other pathways, we don't have like their global locations. And I put a proposal in, I think 4 times now, to develop this model out to include that stuff. But NSF thinks it's not important.
Thank you.
Yeah, so because your first picture shows the Southern River organics, and with my co-authors, we studied Southern River humic acid structure.
Yeah.
And we found this very complicated but very exciting. So, for example, I like to discuss with you for one thing is we found there's a direct interaction between Southern River humic acid and phosphate, because a lot of people use phosphate as buffer for game study. It's easy to punctuate pH at near neutral. So we know that's buffering capacity of phosphate. However, so because people think that negative charge, they don't talk to each other. Our study definitely shows there's a strong direct interactions between DOC and phosphate. So I just wondering, so if that's the case, like for your study, would that affect like for example, those like very constant and the thermodynamics in your system? Because you are talking about organic carbon. But what if there's interaction, direct interaction between organic carbon and phosphate?
So this is again a global study.
Yes.
So we had to take several steps back and say what can we get global information on and incorporate into a model. So we don't go into that level of nuance at all.
Yes.
And the parameters for our model for the rate of organic carbon degradation
Yes.
are very much just looking at what is the rate of organic carbon. That's it. It's not looking at why is it degrading that way or what's in that system, or you can have a complicating factor here or there. And another thing I'll reiterate is that this is a 1/4 of a degree by 1/4 of a degree grid system. So we're looking at, if you're near the equator, it's hundreds of square kilometers. So a lot of times I give a presentation like this and people say, like, well, hey, I study this location over here, and your model does not replicate my location. I'm like, your core is this big. I'm looking at the entire planet. I'm not going to be able to resolve your field site in a global scale model. It's about getting the sort of like large scale trends and quantifying like, what's happening everywhere sort of. And there's plenty of reactive transport modeling at specific locations where they can take all kinds of other factors into account. But we can't, this was not intended to like look at, how does phosphate bind organics or anything like that. Yeah. There's another question somewhere. Yeah.
Thank you, that was an incredible talk.
Thanks.
So you brought up this topic about that most of life is like under the subsurface, right? Or unicellular life at least. So my question is, on the origins of life, we're focused about like sources of energy to be able to like form life. But you bring up this point that the limits of life are in the zepto-watt range. So do you think that would be enough to like spark first origins of life under the surface? Or would you need like an external source and then life kind of like spread down the surface from there?
Yeah, well, the nice thing about the subsurface is that whatever period in Earth history where it's difficult living on the surface due to radiation or a faint sun or impacts. The subsurface doesn't see any of that, it doesn't care. Mostly, I mean, when the moon forming impact happened, yeah, it was a problem. But, you know, smaller impacts, you know, you go down a kilometer or so, it's not a big deal. You don't have to worry about all the surface stuff. So, and especially before oxygen was in the atmosphere, before we had the ozone, you get V coming down, so you're protected at least. I don't really care where it started. It definitely spread down there. It seems pretty well adapted to living in the subsurface. Yeah.
So when you're approaching these femtowide type systems, are the cells just maintenance metabolism? Is there any evidence for actually growing generation times years?
Yeah, so people have tried to calculate generation times based on a number of different things like amino acid racemization. They've gotten initial numbers in the tens of thousands of years. So this is for super shallow stuff. This is not the deep zeptowatt area. And they've kind of revised that to be at least many decades or hundreds of years for a doubling. It's a potential doubling time. They're mostly repairing parts. And for these zeptowatt systems, I think they're just dying slow. They're not really living. Because the cell numbers are going down with that. So there's debt, community-wide debt, for sure. And actually, I have another paper on necromass. You can eat your dead relatives for a while. That'll propel your community for a good bit of time. Hey, when you're hungry, right? But yeah, I don't think hardly anywhere, except those topmost sediments, do you have any go for
Viruses.
So the virus numbers tend to be very low. So this means there's probably not much biological activity. You don't have that standard 10-to-1 sort of number. It's just not much.
I should probably know the answer to this, but I'm sure the concentration of the organics is higher in sediment than in the overlying matter. But if you just did the total mass organic matter in the overlying seawater column versus in the sediment, where is the total mass greater of organic matter greater? And also I assume the composition is somewhat different of the zeptowatts would be different between the organic matter from water column versus the sediment.
Yeah.
Organic matter.
Yeah. So yeah, I don't know at what depth you have to integrate to get the same mass of organics. I don't know. That's an interesting question. Of course, the POC content of the sediment water interface varies by orders of magnitude. You're out in the abyss and it's almost nothing, and you're on the coastal shelf and it's just packed full of goo. But in terms of what was the zeptowatt question? It was about the type of the organics.
Yeah.
Yeah. So the way our model worked is that the water depth helps indicate the initial reactivity of the organic carbon. So if a fecal pellet falls through 5 kilometers of water, it's being degraded all along the way. And when it gets to the sediment water interface, it's not very reactive. That's how our model treats it.
Right.
So that's why we use bathymetry to parameterize that. And so if you're only going through 100 meters of water, then you're more reactive when you hit the sediment water interface. So that stuff is going to react faster. Well, actually, what it has a greater distribution of more reactive compounds because we're using this reactive continuum model. So there's a whole spectrum of reactivity. So you have more of that reactive stuff if you haven't gone through as much of the water column.
The definition DOC at 800 meters versus at 4,000 meters versus at 2 meters depth in the sediment. What?
The oxidation state of it?
Yeah.
I don't know. Yeah, I mean I want to measure these things.
Thank you.
Yeah.
On the plots of relative rates of CO2 and methane production, I was sort of struck by the sort of like initial production time before the methane really picks up. Is the other community of carbon oxidizers, are they like pre-processing the organics or something in a way that like the methane just can't take advantage of it until they've got enough acid edible stuff? Or what?
I don't know what so let me show you real quick. It's bonus slide. Let's see, some other stuff I was gonna say, took all this stuff out. So here are, this is at metagenomes. And so this is giving them a bunch of low oxidation state organics. And this is, gene presence or genome abundance. And so you see all these guys that specialize in the low oxidation state, and all these guys specialize in this high oxidation state. And I don't know what that distribution was like initially before the experiment started. We looked at it afterwards, right? So there's low oxidation state ones, definitely these became very abundant, and then they get the high oxidation state, and these guys all became very abundant. So if the initial distribution was for the specialty of high oxidation state, then yes, it would make sense that they are faster at processing organics because the high oxidation state specialists were already in a high abundance. I don't know that though, unfortunately. I don't know what it started out as. And I don't know. So the plot this came from is one of these experimental plots, a rice paddy plot in California. And I don't know what they add for fertilizer. That would be another fun question to answer, to see like how maybe the community is just ready for higher oxidation state material. So that's why, possibly. I don't know. But we do think that because they get less energy from these lower oxidation state compounds, maybe they haven't evolved to process them quickly because they're always trying to get those high oxidation state things that are more energy. But yet it's different communities doing it, so that doesn't quite make sense either. So I really don't know, and I'm just talking out my other side right now. Roy.
So yesterday we heard from AP and geosciences talking about biosignatures and organics preservation potential on various sites around the Earth as an analog to Mars and to ancient Mars, which has been sort of in stasis for 3 billion years. She's finding organics everywhere. They find it everywhere. And so my question is, like, in terms of preservation potential, we'll talk about a lot of lipids. Lipids are being preserved, and that's what we use as a biosignature here on Earth for ancient life. Is that a function? Like, to me, a lipid is still something that a microbe would love to eat.
And they do. We don't walk around on a planet full of 20 meters of lipids.
Right. And so my question is this prevalence or long-lasting existence of the lipids, for example, as one example, is that because they can't eat it because they're lacking an electron acceptor, or is that an energy requirement that they can't reach, or why are the lipids still here? Right.
So
In some places and things.
there's another plot in this paper that I don't have in the slide deck, but it is showing what I've called the thermodynamic limiting factor. So the idea is that when reactions get lower and lower in energy, their rates slow down. This is known. This is not me making anything up. But the question is, does this apply to microbes eating organics? You take any other reaction, and as it gets close to equilibrium, its rate slows. And in fact, it can never get to equilibrium. It's an impossible situation. We just approximate it. So the idea is, in an anaerobic system, when you have these low oxidation state compounds, they give you a lot less energy. So the rates approach zero. So it could be that you are in a system where you're getting so close to equilibrium because your oxidants are in low supply and your organics don't give you much energy. There just isn't a threshold that can't be overcome in a preserved stuff. I was a part of a paper audit a dozen years ago where we looked at just lipids and sediments and looked at the preservation of them. And it turns out that it's scaled with the oxidation state level, not the sea surface temperature or all the other stuff that people use lipids for. I'm sorry I'm digging on lipids right now, but microbes like to eat stuff, right? And I like to eat these organics, and they're supposed to tell us about paleo environments. And sometimes I think, well, maybe they just couldn't eat them. And that's why they're still preserved, not because more of them were produced at the sea surface, because it was warmer 5 million years ago. It's because they can't. And so that's why they're still around, not because they were more produced. So I don't know that to be the case, but at least in one environment, we showed that the surviving lipids scaled with the oxidation data, but not some other, climate parameter.
So it all has to do with the oxidation zone.
Wow, I would say all. It's a telling diagnostic feature of systems. Be nice to look at.
All right.
All right, thank you so much.
Thank you.
There's somewhere around